Okay, let's unpack this. We are diving into the world of infection control and asepsis today.
Yeah, it's fundamental.
It really is. And our sources, they highlight this kind of paradox, don't they? That
Yeah.
the simplest, lowest tech actions are often the most critical.
Absolutely. And sometimes the hardest to get right consistently.
That seems to be the key, consistency.
It is.
Yeah.
There's this story about a labor and delivery nurse, Jarek Schvetz.
Oh, right. I remember reading that.
So he'd had this incredibly busy day, assisted with 5 births, resuscitated an infant, really intense stuff.
Yeah.
And later, his daughter Stephanie asked him, "What was the most important thing you did today?"
And you'd think he'd mention the resuscitation or one of the births.
Exactly. But his answer, he said, "I wash my hands a lot."
Wow. That says so much, doesn't it? About priorities, about what really matters on the front line. Simple diligence.
It's the bedrock, especially when you look at the stark reality of healthcare associated infections, or HAIs.
Yeah, the stats are pretty sobering. HAIs are the leading complication of hospital care.
That's right. And incredibly, one of the top 10 leading causes of death in the US. It's a huge, often silent threat within healthcare settings.
And that number, approximately 1 in every 31 hospitalized patients gets at least one HAI.
Yeah, about 1 in 31. It just reframes everything, doesn't it? Shifts the focus from, complex surgeries to just basic constant awareness and action.
So that's our mission for this deep dive then. Let's really dissect how infection actually happens.
Understand the body's own defenses.
Right. And uncover these critical practices like hand washing that break that chain of transmission, even when they're challenging to maintain.
Sounds like a plan. To stop an infection, you really have to understand its journey.
Okay, so this is where we get into the chain of infection. I found this part really interesting. 6 links, right?
Exactly. 6 links. And crucially, all 6 have to be present in sequence for an infection to actually be transmitted.
So if you can break just one link
Yeah.
anywhere along the chain.
You stop the transmission. The cycle ends right there.
Okay, link 1.
Link 1 is the infectious agent, the pathogen itself. So we're talking bacteria, viruses, fungi, protozoa, that sort of thing.
The bad guys, basically.
Well, yes, but it's also important to remember our normal flora, the microbes that normally live in and on us.
Right, like in the gut, helping with digestion.
Precisely. They help digest food, synthesize vitamin K. They're beneficial in their normal place. But if they get somewhere they shouldn't be, like E. coli from the gut ending up in the urinary tract.
Then they become the infectious agent, a pathogen.
Exactly. Location, location, location.
Okay, so we have the agent. What's next? Link 2.
Link 2 is the reservoir. This is simply the place where the pathogen can survive, multiply, and hang out waiting for a chance to spread.
What kind of places are we talking about?
You generally prefer environments that are, warm, moist, and dark.
Makes sense.
And reservoirs can be living things like humans who might be sick, or carriers without symptoms, animals, insects.
Or non living things.
Yes, non living reservoirs are everywhere: soil, food, contaminated water. But in healthcare, think about things like sinks, bed rails, bed linens, even equipment.
And the source material had that really stark example, didn't it? About the patient bath basins.
Oh, yeah, that was eye opening. Yeah, a study found, what was it? 98% of tested bath basins in hospitals grew bacteria.
Including things like MRSA. So the very thing meant for cleaning.
Can become a primary reservoir itself.
Yeah.
Which leads us to link 3, the portal of exit.
How the bug gets out of the reservoir.
Exactly. Think bodily fluids, coughing, sneezing, saliva, diarrhea.
Okay.
Or it could be drainage from a wound or exiting through tubes like IV lines or catheters. Basically, any way out.
Got it. So the agent is out and needs to travel. Link 4.
Link 4. The mode of transmission, how it gets from the reservoir to the next person.
And there are different ways this can happen.
Primarily, yes. The most common is contact transmission.
Like touching someone.
That's direct contact, skin to skin. But there's also indirect contact. This involves an intermediate object, what we call a fomite.
A fomite. Like a contaminated object.
Precisely. Think of a stethoscope, a keyboard, a doorknob, that bath basin again. The pathogen gets on the object and then someone else touches it.
Okay, so contact is big. What else?
Then there's droplet transmission. This is when someone coughs or sneezes and moist droplets carrying the pathogen travel, usually just a few feet, and land on someone's eyes, nose, or mouth. And finally, airborne transmission. This is different from droplets. These are tiny microorganisms that can float on air currents for considerable distances.
Like TB or measles.
Exactly. And sometimes these could be stirred up just by, say, shaking out contaminated bed linens. They just hang in the air.
Okay, so the pathogen has traveled. It needs to get in. Link 5.
Link 5 is the portal of entry, the pathway into the next host.
So, like the mouth, nose, eyes.
Yeah, normal body openings are common entry points, but also breaks in the skin, like cuts or scrapes, surgical wounds, or insertion sites for needles or tubes. Anyway, in.
Right. And the final link, number 6.
The final link is the susceptible host. This is a person who is actually at risk of getting the infection because their defenses aren't adequate to fight it off.
Who tends to be more susceptible?
Well, think about the extremes of age, the very young and the very old.
Their immune systems might not be as strong.
Right. Or people who are already immunocompromised, maybe due to chronic illnesses like diabetes or malnutrition or certain medications like chemotherapy or steroids, their defenses are lowered.
So, all 6 links: agent, reservoir, exit, transmission, entry, susceptible host. They all have to line up perfectly.
Every single one. Break any link and you prevent the infection.
Okay, that framework is really clear. But, the challenge today seems amplified by these evolved invaders, the MDROs.
Ah, yes, multi drug resistant organisms, NDROs. This is a huge, huge issue.
Explain that a bit more. What makes them so challenging?
So these are microbes, mostly bacteria, that have essentially mutated. They've developed resistance to one or more of the antimicrobial drugs, like antibiotics, that would normally use to treat them.
So the treatments don't work as well or at all.
Exactly. This severely limits our treatment options, and the consequences are serious.
Wow.
More severe illness, higher death rates, longer hospital stays, and just vastly increased healthcare costs.
What are some of the main MDROs that hospitals are really grappling with?
Well, two really prominent examples come to mind immediately. First, MRSA, that's methicillin resistant Staphylococcus aureus,
Heard of that one. It's spread by touch.
primarily, yes. Skin to skin contact is a major road. And the problem is, MRSA can survive quite well on surfaces and equipment for a while.
Okay. And the second one?
The other big one is Clostridioides difficile, usually called C. diff.
Right, C. diff. That's often linked to antibiotic use, isn't it? Especially in older adults.
Very much so. Taking antibiotics can disrupt the normal gut flora, allowing C. diff to overgrow and cause pretty severe diarrhea and colitis.
And C. diff has a particular trick up its sleeve, doesn't it?
It does. It forms spores. These are like dormant, highly resistant versions of the bacteria. And these spores are incredibly tough. They can survive for days, even weeks, on surfaces like doorknobs, bid rails, toilets.
Which brings us to a really, really critical point about cleaning, specifically hand hygiene.
Absolutely crucial. This is a major practice point. Standard alcohol based hand sanitizers. They work great against many bacteria and viruses in their normal vegetative state,
But not spores.
but not against spores like C. diff. Alcohol doesn't effectively kill the spores.
So if there's any risk of C. diff contact,
No.
maybe caring for a patient with it, what must you do?
You must wash your hands with soap and water. The physical friction of washing, rubbing your hands together vigorously with soap and rinsing with water is what actually removes the spores from your hands. Alcohol rub won't cut it.
That distinction is vital. Soap and water for potential spore contact. Okay.
Non-negotiable.
All right, let's shift gears a bit. We've talked about the invaders and the chain. What about our built in defenses, the body's own immune system?
Right, before we even get to medical interventions like hand washing, our bodies have their own defense system working constantly. We usually think of it in 3 lines.
3 lines of defense. Okay, what's the first line?
The primary defense. Think of this as our outer wall, our structural barriers. It's non-specific, meaning keeps everything out, not just particular germs.
Like our skin.
Exactly. Intact skin is a fantastic barrier.
Mm-hmm.
Also, mucous membranes lining our airways and digestive tract, they trap pathogens.
What else?
Things like the acidic environment in the stomach, which kills many ingested microbes. Enzymes like lysozyme in our tears and saliva have antimicrobial properties.
Hmm.
And even our normal flora, competing with invaders for space and nutrients.
So a whole range of barriers and general defenses.
Correct. That's the first line.
But what if something gets past that? What's the second line?
If a pathogen breaches the primary defenses, the secondary defense kicks in. This is still non-specific, but it's more of an internal biochemical response.
Kind of like inflammation.
Inflammation is a key part of it, yes. When you get a cut and it gets red, warm, swollen, and painful, that's inflammation at work.
How does that help?
Well, the body increases blood flow to the area, makes the capillaries more permeable. This rushes specialized white blood cells called phagocytes to the scene.
Phagocytes.
Yeah,
They eat the invaders.
essentially, yes. Phagocytosis is the process where these cells engulf and destroy pathogens and cellular debris. Inflammation helps get them there quickly, and walls off the area.
Makes sense. What else is part of the secondary defense?
Fever is another one. Raising the body's core temperature can inhibit the growth and multiplication of some microbes, and it can also enhance certain immune responses.
So, inflammation, phagocytosis, fever, the body's immediate reaction force.
A good way to put it. Still pretty general, though.
Okay, so what's the third final line? This must be the specific one.
Exactly. The tertiary defense is our specific adaptive or learned immunity. This is the highly sophisticated part of the immune system that recognizes, targets, and remembers specific pathogens.
This is where things like antibodies come in.
Precisely. This involves specialized lymphocytes, T cells, and B cells.
Mm-hmm.
B cells produce antibodies, which are proteins that lock onto specific invaders, marking them for destruction.
Like putting a target on them.
Exactly. T cells do various things, including directly killing infected cells, cellular immunity, and helping to coordinate the overall immune response.
And this is the defense that gives us long term protection, right? Why we often don't get the same illness twice.
Yes, it creates immunological memory. The system remembers the pathogen, so if you encounter it again, the response is much faster and stronger, often preventing illness altogether. This is also the basis for how vaccines work. They stimulate this specific learned immunity.
So, primary barriers, secondary rapid response, and tertiary specific memory. Quite a system.
It's incredibly complex and effective.
Which makes it even more important to support it with external measures, like medical asepsis.
Right. Medical asepsis, often called clean technique, refers to practices aimed at reducing the number, growth, and spread of microorganisms. It's about proactively breaking that chain of infection we talked about.
And it's generally simple stuff, right? Standardized, relatively low cost.
Mostly, yes. It involves things like proper cleaning of the environment and equipment using personal protective equipment,
Mm-hmm.
but the absolute cornerstone, the single most important activity.
Has to be hand hygiene. Back to Jarek the nurse.
Absolutely, hand hygiene. It's recognized globally as the first and most critical patient safety challenge because it's so fundamental, so effective, yet, as we've noted, it's so challenging to do perfectly every time.
Let's really break down what makes hand hygiene effective because just running hands underwater isn't enough.
Not at all. There are key factors. First, time.
How long?
If you're using soap and water, the recommendation is vigorous scrubbing for at least 15 seconds. Cover all surfaces.
Okay, 15 seconds minimum, soap and water. What about alcohol rubs?
For alcohol based hand rubs, you need to apply the recommended amount and rub it over all surfaces of your hands and fingers until your hands are completely dry. This usually takes around 20 to 30 seconds. Don't wave them dry or wipe them off.
Rub till dry. Got it. And you mentioned friction earlier, especially with soap and water.
Crucial. You need to rub vigorously.
Yeah.
Get between the fingers, rub the backs of the hands, the palms, and pay special attention to the fingertips and underneath the fingernails. Those areas are often missed and can harbor a lot of microbes.
Under the nails, right. Easy to forget.
Very easy. And the third key factor is agent specificity, choosing the right tool for the job.
This goes back to the C. diff spores.
Exactly. For routine hand cleaning, when hands aren't visibly soiled, a 60% or higher alcohol based rub is usually preferred. It's effective and often quicker and less irritating than soap and water. But if your hands are visibly dirty, or if you've potentially come into contact with spore forming organisms like C. diff, then alcohol rub is not sufficient. You absolutely must use soap and water and friction to physically remove those spores.
Time, friction, agent specificity seems straightforward.
It does seem straightforward.
Yeah, the compliance isn't always there. You mentioned the challenges.
Yes. Despite very clear standards from organizations like the CDC and the Joint Commission, which focus heavily on reducing specific HAIs like central line infections or catheter associated urinary tract infections, research consistently shows that healthcare staff don't always follow the guidelines.
Why not? Is it lack of knowledge?
Generally, no. Most healthcare workers know what to do and why. The barriers identified in studies are often systemic.
Like what?
Things like excessive workload, being understaffed, feeling rushed.
Just too busy?
Too busy, yes. And sometimes practical things like sinks or hand sanitizer dispensers not being conveniently located or running out of supplies like paper towels. These small barriers add up.
But the consequence of noncompliance is huge.
It's enormous.
Yeah.
From a professional standpoint, failure to perform known standards of care, like proper hand hygiene, that results in harm. Well, that constitutes medical negligence. And beyond the ethical and professional implications, there's a significant financial one too.
How so?
Institutions can be penalized. For instance, Medicare often won't reimburse hospitals for the extra costs associated with treating certain preventable HAIs if they occurred during the hospital stay.
So the hospital essentially eats the cost of a complication that might have been prevented by better adherence to basic protocols.
That's the reality. It puts immense pressure on facilities to ensure compliance, but as we said, the barriers are often tied to resources and staffing levels, those tricky human factors.
So we've gone full circle. From the complexity of the infection chain, the scary evolution of MDROs, the body's own intricate defenses.
And we land right back at the simplest, most fundamental action: washing your hands correctly and consistently.
It really highlights that human diligence, that consistent attention to basic detail, is the ultimate linchpin in biological safety, doesn't it?
It truly does. The success of the most advanced medical procedures can hinge entirely on whether someone remembered to wash their hands properly for 15 or 20 seconds.
It makes you think, if something as basic as hand hygiene can be compromised by systemic issues like workload and staffing, what does that tell us about our healthcare priorities?
It's a critical question.
And it forces you as a patient or just as a member of the community to think about those human factors.
Mhm.
How much does the pressure on healthcare workers affect the safety protocols that protect all of us? And how do we translate that vigilance into our own lives? Using sanitizer, not touching our faces, keeping distance when needed to limit spread outside the hospital, too.
That vigilance, both institutional and personal, seems to be the enduring lesson.